Good morning and good evening, everyone. Welcome to today's Stem Cell, Organoid, and Regenerative Biology seminar. And this is the Asia and Australia session. This meeting is part of a series of online seminars organized by the Human Cell Atlas Developmental Biology Network. We try to take the opportunity using these seminars to think about creating an atlas of human development and what is it for, why do we do it. Of course, there is always the fundamental interest in just how the human organism develops, which is more than enough justification in itself. But there are also many applications where a deep understanding of developmental processes is crucial to human, for example, disease research. And not least in the stem cell and the regenerative biology fields, where developmental mechanisms repeat themselves in the adult organism. And organoids in particular provide access to tissues that are impossible to even obtain from living humans, and allow manipulations that are that cannot be done in vivo in humans. So, for example, the earliest stages of human development or the development of the human brain. And so therefore today's theme is very timely and topical, and we look forward to an interesting set of talks and a panel discussion. We're using Slido, so in order to submit questions and vote for questions for the panel discussion, please go to slido.com/HCA dev, and then once you're there, click on Q&A to submit your questions, and you'll be able to vote for them as well. We also like you to share resources or ideas, suggestions, and so you can do that using the Ideas tab. To kick us off, we have polls. So if you go to the polls tab, you'll find a few questions. We'll start with this one. How did you hear about this seminar? I'm going to leave you about 30 seconds to get there and start voting. So we'll see the counter ticking up there, I think. Yes. And if you're having trouble finding Slido and logging in, I think there's also information in the chat on the Zoom that you can refer to later during the talk. Right, I see people getting into Slido. That's great. Nobody uses Twitter or Facebook. It's fine. The second poll, we'd like you to share what you'd like to get out of this seminar. Again, go to the polls and you'll find this question, and you can type in your answer, and we'll get some suggestions here on the screen. Okay, that's great. I think let's go ahead with the seminar. We have a great lineup of speakers today. We are going to hear 2 lightning talks by Barbara Treutlein and Carl Köhler, and then our Australia speaker, Christine Wells. We can look forward to 3 interesting talks on the theme. But please, during the talks, keep submitting your questions to Slido and vote, and then we'll have a panel discussion. We hope to have a lively discussion about all these topics. So let me first introduce Barbara Treutlein. She's from ETH Zurich in Basel, and has in 6 very short years gone from being a postdoc at Stanford to becoming one of the most influential researchers in the field of single-cell biology. And her talk today is titled Enhancing the Human Cell Atlas with Human Organoids. Go ahead, Barbara.
Great, thank you very much, Stan, for this kind introduction. It's a pleasure to be here and part of this set of speakers and then discuss in the panel. I'm very much looking forward to that. So I will tell you a little bit my perspective in a short time, how we can enhance the human cell atlas with human organoids. And I go right in. You see here really some examples of organoids that we have grown in the lab, and these are really amazing. And it is a really exciting time to study human development, because we have these in vitro systems available where we can, from any individual, reprogram somatic cells into induced pluripotent stem cells. We can then differentiate those into different lineages. And the most exciting to us is that they can then also in 3-dimensional culture differentiate into different human organs in vitro. And so we can grow these organoids from induced pluripotent stem cells, also from adult stem cells. We are very much focusing on these iPS-derived organoids, which mimic human development, whereas this other path is more a mimicking of tissue maintenance and regeneration. And the exciting part is that these organoids can be grown from any individual, such that they are avatars for healthy organ development, but also for what can go wrong in disease and modeling disease in vitro. And so I said this already, so these organoids are specific to an individual. We can do time course measurements, so we can actually, and Stan mentioned this, we can fill in times that we cannot measure based on primary tissue because the tissue is not available. We can manipulate these systems, we can control their environment, and they are amenable to high throughput screening. So overall, I think these organoids can enhance the human cell atlas by complementing these primary cell atlases. I want to show you in a very short fashion how we imagine to do this based on brain organoids. So these are these cerebral organoids that grow from embryoid bodies within months, actually. And here you can see a 1.5-month-old organoid, a cross-section through it, where you can appreciate that, for example, regions develop such as this one here that resembles the developing human cortex, but there are also other regions. And so using high-throughput single-cell transcriptomics, we can now, for example, track really the development from pluripotency into different brain regional identities, such as the cortex, the ganglionic eminence, mid and hindbrain. And we see not just neuronal cells, as are shown here, but also astrocytes and supporting mesenchymal cells. We can grow these organoids also from different individuals. So here showing just 7 different lines and an integrated network of all this data. And then we can, within a given trajectory, for example, compare across the individuals, how does expression of given genes vary across the pseudotime. And here we can then identify potential inter-individual variation. We can also go into technical aspects and see how different lines behave differently or give different cell proportions in the final organoid and really assess the heterogeneity of these tissues. Now, as I said, these are genetically manipulable, so we can manipulate cell states and understand function of genes, and this is really not possible with primary tissues. And just as one example here, we, for example, knock out a transcription factor in iPS cells, and we have control cells that went through the same process. And when we then perform single-cell RNA sequencing on these organoids, again, we see different trajectories appearing. So these are 2-month-old organoids. We see, again, the cortex, the ganglionic eminence, and these other brain regional identities. And very interestingly, for example, in this case, this transcription factor knockout leads to really a loss of the dorsal telencephalic lineage. So we can really identify how important transcription factors are during certain fate decisions. What we can also do with these in vitro systems is to really directly trace lineages. And so one thing that we developed, this is available on bioRxiv, is, for example, a way to introduce barcoded GFP into iPS cells. So every iPS cell has a lineage mark based on this barcode, unique barcode. But then we can also, at any time point of interest, we can induce Cas9 expression, which will lead to a scarring of the GFP based on this guide RNA that is also present on the same iTracer vector. And in this way, we can then introduce a second lineage mark through scarring of GFP. And using single-cell RNA sequencing, we can then identify all the cell states that are present based on the transcriptomes. But based on the combination of these barcodes and scars, we can reconstruct a lineage tree of all the cells that we detected. And we can identify barcode and scar families. And this is, of course, particularly interesting when now comparing a wild type with a diseased or manipulated scenario. What we can also do with these tissues is actually they are amenable to time-lapse microscopy. Here, a light sheet movie, a cross-section through an organoid that we image in 3D over many days. And you can see how these early events happen where these lumen form, and we can use this data to track individual cell lineages. So now not just inferring them using these scars and barcodes, but actually directly track cell divisions and how cells contribute to the tissue. And so my last point would be that we cannot just enhance the human cell atlas by using organoids. We can actually also enhance organoids by using the human cell atlas. So this is a two-way thing. Here, when we have all these organoid atlases, and then we also have these primary atlases for matching time points, we can really compare quantitatively the cell states we find in the organoid compared to what is found in primary tissue. And this can help enhance organoids. We might find cell lineages that are missing, such as immune cells are currently missing a lot. We might find off-target cell states in the organoid that are not found in the primary tissue. And with this, we can then reverse engineer the organoid and really try to make these in vitro systems even better. And I want to point out that there's a network, HCA biological network on organoids, where Christoph Bock and I are the coordinators. And so please, if you want to somehow contribute and join the discussion, you can contact us. And with this, I'm at the end. And I want to thank all the people in my group, our great collaborators, and in very particular, I want to thank Gray Camp, who is also in Basel at the Institute of Ophthalmology in Basel, and all these efforts we are pushing together. And of course, also thank the funding. Thank you very much. And now I'm handing back to Stan.
Thanks, Barbara. That was great. We are going to hear our next lightning talk from Carl Kohler from the Boston Children's Hospital, who's doing some amazing work taking organoids to the next level by integrating multiple lineages. And you will see for yourself in this talk titled Mapping Sensory Development in Organoid Models. Go ahead, Carl.
Okay, thank you, Sten, for the introduction, and thanks to the HCA for, again, the invitation to speak with you today. And thanks, Barbara, for the nice introduction to organoids and working with stem cells. So I'm just going to very quickly tell you about our work over the last decade or so, trying to develop some very hard to generate organoid systems. We're looking specifically at sensory systems. And this first slide kind of shows you, and gives you a nice framework of understanding the sensory system. So we're thinking about building multiple lineages into these organoids, starting from pluripotency and generating not only epithelial cells, which in the context of a sensory system, would contain sensory cells. In these three-dimensional cultures, they form these cystic structures with the lumen. And also sensory systems require wiring to the brain. So they require neurons and glia to create the nerve cells, as well as mesenchyme or supporting cells to support these structures. And within the stem cell differentiation field, people are thinking about various ways to get to these complex structures. One way that we've found is that we can actually generate all three of these groups of cells directly from pluripotency within the same aggregate of cells. You can also think about generating each of these individual components by themselves and then combining them together. And that's something maybe we could discuss in the panel discussion. And as Barbara mentioned, there are other missing components to these organoid systems that our group and other groups are thinking about ways that we can co-generate immune cells or vasculature to incorporate into the organoid structures. And then another thing that we're working on in the lab is thinking about ways we can generate assembloid-like structures by generating also the central nervous system targets of these sensory nerves, all within the in vitro environment. So over the last decade, we've generated this kind of sensory organoid platform where we can take pluripotent stem cells, generate an aggregate of cells, and induce surface ectoderm cells, which represent the surface of the embryo. And then we see co-development of cranial neural crest cells, which give rise to a variety of tissues within the cranial region of the embryo, including neurons, glia, pigmentary cells, melanocytes, as well as mesenchymal derivatives. And then we've devised a way to kind of toggle between two different sensory systems, either the inner ear or skin tissue. And I should mention that this combination of surface ectoderm and cranial neural crest is kind of a recipe for the head. It forms a number of different tissues within the head region. So the key insight from the work that we've done is that we can really partially direct differentiation in this three-dimensional environment and stimulate self-assembly of organoids with epithelial, mesenchymal, or epithelial neuroglial and mesenchymal cells. So back in 2017, we published work showing how to generate functional hair cells, the inner ear sensory cells contained within these inner ear organoid structures. So you can refer to that paper to read more about that process. We also saw co-development of sensory nerves of the inner ear that are actually innervating the hair cells within the system and are able to show that the hair cells with their stereocilia bundles are actually functional like native hair cells. And so we're using this system now in the lab to develop with collaborators to develop gene therapies for congenital hearing loss disorders. So another project that we've been working on that kind of spun out of this work on the inner ear is to generate full thickness hair-bearing skin tissue. You can see back on this previous slide that we see co-development of this structure right here, which actually represents full thickness skin tissue. So by removing the signaling cues for inner ear induction, we can actually bias the culture towards generating skin tissue and produce these large cystic structures with an inner layer of epidermis and an outer layer of dermis that over time, after approximately 70 days in culture, we see this outward growth, this budding of hair follicles growing outward from the skin organoid structure and nicely mimicking the normal development of human hair in the embryo or in the fetus. Just a few more images of these skin organoids, we see, again, co-development of sensory nerves with the skin organoids. And we see innervation of the hair follicles at the sites that we would expect them to be innervated. We can implant the skin organoids into a nude mouse model. And remarkably, we see kind of opening up of the cyst and integration into the skin. And formation of fairly normal looking human skin islands within these xenografts. So we're really setting the stage for investigation of a number of different topics: human development, congenital skin diseases, skin immunology, and cancer. So referring back to the HCA, one thing we were doing in the lab is doing kind of a single cell RNA sequencing survey of human skin development using our system. We've already published several time points at day 6, 29, representing these different fetal weeks. And we're expanding on that and getting higher and higher resolution, and have recent collaborations with HCA group members to compare and contrast our organoid systems with human fetal skin development. And so in that way, we're thinking of the organoid systems as a nice way to fill in the gaps of pediatric developmental maps, where we're somewhat reliant on very rare and precious samples of human fetal specimens. And so we think the organoid systems that have heterogeneity and complexity, like our skin and inner ear models, could really contribute to building these more higher fidelity maps. So with that, I'll thank my group and pass it on to the next speaker. Thank you.
Thanks, Carl. Our next speaker is a full talk from Christine Wells of the University of Melbourne. She's a professor of stem cell systems biology and a director of the Center for Stem Cell Systems. And her research focuses on the intersection between innate immunity and stem cell biology. And we're going to hear a talk today titled, "Could stem cells provide a roadmap for diversity in the Human Cell Atlas Developmental Atlas?" Please, Christine.
Thank you very much, Stan, and thank you to the organisers for the opportunity to speak to you today. It's great to be participating in a HCA meeting in our time zone. I really appreciate it. So I have 2 parts to my talk. The first is just to elaborate a little bit on how we might apply organoid and stem cell biology to tackling some of the really important issues for HCA in Asia and other countries which may have emerging scientific challenges that aren't equivalent to the challenges that you see in Australia or US or the UK and Europe. And then the second part of my talk, I'm just going to speak to some of the challenges of using organoid technology, but from a computational stem cell biology perspective. So I very grandly said that I wanted to speak about equity and whether organoids could address an equity issue within the Human Cell Atlas project generally and within HCA Asia specifically. And I think it's important to note that there have been a lot of big genomics projects which simply haven't included populations outside of the US and Europe. And it's a really important equity issue because inclusion of diversity of genomes and inclusion of diversity of people and ideas makes our science better and makes the impact of our work more relevant. But there are lots of barriers to having equitable access to consortia activities like the Human Cell Atlas. And beyond the opportunity to contribute, there's also this need to build a framework of training, infrastructure, ethics, engagement and policy activities, and capacity for clinical translation on top of the research activities that really allow you to be fully translational and fully committed to outcomes from a stem cell perspective, and I think from a global genomics perspective as well. I think that there are particular challenges when you're working with low to middle income countries in terms of distinguishing between capability and capacity. Capacity often just comes down to funding. There are often quite different regulatory frameworks in the lower to middle income countries, and even within our region, different regulatory frameworks to deal with people who come from, for example, Indigenous backgrounds compared to Caucasian backgrounds here in Australia. I think it's critical that we recognise leadership beyond the kind of traditional academic metrics when there are big barriers to overcome to participate in these global projects. Then leadership comes in many different forms, and it's really important to recognise the efforts that are already underway for HCA Asia in particular, and to support those, particularly because within our region, working across borders will be necessary to consolidate research outcomes. So I'm really excited that HCA is branching into organoid biology, because I think that organoid biology has got an opportunity to address many of the equity and access issues that we are struggling with at the moment. For HCA Asia, a lot of the activities are focused on sampling blood, but blood, of course, has got a finite life in the lab, and so there are other things that we can do if we transform those blood cells into iPS cells that allow us to reach a more clinically relevant outcome for the populations that we're working on behalf of. And here is an example in cardiac biology where the use of cardiac organoids can rapidly eliminate a safety burden for new drug development, where there may be unexpected cardiac toxicity effects that require very invasive tests to identify, but which can also now be picked up using cardiac organoids. And indeed, in the US, the FDA and others are starting to take advantage of screening capabilities in cardiac organoids to test for potential drug side effects before these come onto the market. Now, one of the key points in the HCA Asia activity is the point that there are a lot of diseases, but particularly cardiovascular diseases, which impact on different Asian populations in quite a distinct way compared to Caucasian populations. And so being able to screen drugs within the population that you want to distribute that drug in is a really critical equity issue. We also know that if we're interested in really understanding population genetics and the contribution of, for example, expression QTLs to the phenotype in a dish that we're looking at, that blood can only get us so far. And this was a very influential paper for me, thinking about this problem from Job Gillard's lab, where he took, again, cardiac organoids and explored the relationship of QTLs with expression changes over a 15-day differentiation time course. And the three graphs I'm showing you really say the same thing, and that is that it's in the first 5 days of that differentiation protocol that you see the really strong expression QTL drivers that are informative about the behaviour and the formation of the tissue. And that if you wait and look just at the mature cardiac tissue, then you may as well be looking at blood in many respects. So organoid biology allows us to get a window into that time in early organoid patterning where genetics may have quite a profound effect on later life. So just to summarise why I think organoid biology might provide a really important model system from an equity perspective in the HCA is that it does provide us with this opportunity to reveal expression QTLs which are associated with the development and function of major organs. It also provides a model system for rapid functional validation and potentially for clinical action, particularly if that clinical action is linked with the capacity to screen either new candidates for disease treatment, but also ensure safety and efficacy of drug targets coming into the country. And the lack of genetic diversity in organoid biology in this respect, I think, is a really important equity issue for us all. I also think that from an ethics perspective and an inclusion perspective, derived cell types may be more acceptable to cultures who have objections to sharing body parts, either unable to share cadaver material or unwilling to donate blood but willing to donate DNA and RNA, for example. In Asia, there is a particular research strength in stem cell biology and organoid biology, and certainly there are existing major investments in Japan, Korea and Taiwan for IPS banks that we could be leveraging from. So I think that there is a strong opportunity for contributions to the HCA organoid program to contribute not just to our understanding of how organs form, but also what the impact of different genetic backgrounds have on the formation of those organs, and then a really important knock-on effect in providing a platform for equity access to clinical translation in this area. So just moving on to part two and talking through some of the challenges of using the technology, and particularly from a computational stem cell biology perspective. So here I'm showing you two images. On the left, you can see a fish. This is a principal component analysis of all of the pluripotent stem cell data that we host within the StemFormatics platform. And on the right, you can see a t-SNE plot of the Human Cell Atlas cord blood data. And the point behind these two figures is that we rely very heavily on these images to draw relationships and ideas between the cells that we're visualizing. But if you get the primary data transformation steps wrong, then your eye is often drawn to patterns within those data sets which may not reflect the biology at all. But because we are very visual species, we will develop stories about these clusters regardless of whether or not they can be, whether or not they reflect anything valid at all. So data integration is a really critical part of the Human Cell Atlas pipeline, and in particular when you're using organoids or stem cell biology, benchmarking the similarity of the in vitro derived cells with their in vivo counterparts is really important. And there are three different types of approaches that you can use to get to how similar or different your cells are. So the most common approach in the single cell world is to harmonize data sets. And that is where you take one data set as the ground truth and you anchor the other data sets to that. And it's a very effective way of identifying categories of cells which are present in the reference but absent in the cell set that you're looking at. And when we apply this to organoid biology, we often see that there are some tissues which simply aren't included in our organoid models. The second approach that you can use is to normalise the data, and this is essentially where you are choosing which categories you want to compare, and you are rescaling the data so that those categories meet the statistical assumptions of the model that you're looking at. We lose a lot of data, particularly biological variants, when we apply harsh normalisation, particularly sparse data like single-cell data. And then the third approach, which is not used very often at all, is the approach of variance filtering. And this is where you remove genes that have got a big impact from technical artefact, so that you're only looking at the behaviour of the cells from the perspective of genes which are reflecting the biology you're interested in. And although you may lose a large number of gene features from this approach, you often reveal new variables which are influencing the behaviour of the biology you're interested in, such as new attributes of the donors you're looking at. So I think it's really important when you're looking at organoid biology in particular that you have a heuristic which lets you explore all three types of data integration methods, so that you're not just relying on harmonisation to benchmark your cells, but you're also allowing for exploration of other variables in the data. Just to show you that this variance approach is very effective, if you have a look across all of the genes that you might be needing to normalise in an integration experiment, what you can see from this graph is that the majority of them actually have got some proportion of variance explained by the way they were measured, the sample source or the platform, but quite a large number of genes have got a very low platform effect and can be attributed entirely to the biology that you're measuring. So if you treat every one of the genes here the same way and apply the same normalisation scale to it, then you are quickly going to be removing some genuine expression values which can tell you something about the biology you're interested in. When we take this variance modelling approach and apply it to very well-studied systems like blood, here I'm showing you not single-cell data, this is traditional RNA-seq data. You can see that we can filter down to just over 3,500 genes to plot and combine data from about 1,000 samples, which have been sampled across a lot of different platforms. And what can emerge here very readily is the behaviour of common myeloid subtypes within blood. So taking this approach, we're asking what is the impact of culturing cells and how equivalent are cells that you might derive completely in vitro. And here you can see there's quite a big difference, for example, for cord blood-derived dendritic cells, which is a common human dendritic cell model, compared to blood-derived dendritic cells, which I'm showing on the bottom of the screen. If we use this reference as our anchor for single-cell data, and we plot on top of that several different single-cell data sets, we can quickly see how similar those data sets are to samples where we know a lot about their phenotype. And we can also start filling in the gaps between the samples that are hidden in the bulk profiling but revealed in the single-cell profiling. And in this way, we can classify the samples that we're seeing in single-cell data against a reference atlas of blood. And so, for example, using this approach, we can identify 2 different plasmacytoid DC subtypes which have been found within one of the single-cell data sets, but not the other. So my lab is really interested in macrophage biology, and we make macrophages in the lab. They do all sorts of macrophage things. They share all the cell surface markers of macrophages, and they're highly phagocytic. But we applied this question of how similar are these macrophages to the in vivo macrophages, then what we can see is that there are some really pretty big dissimilarities. So on this graph, I'm showing you, again, bulk RNA-seq. All of the in vivo derived cells are sitting at the bottom of the graph, and all of the in vitro derived cells are sitting at the top of the graph. And the pluripotent derived cells are sitting quite a long way away along the third component from the in vivo cells. Now, these include cells from my laboratory, but they also include cells from about 40 other studies from a number of different laboratories around the world. So this is a very consistent phenotype. You can see in pink the hematopoietic progenitor, stem and progenitor cells, which have been taken from bone marrow. And there is a clear trajectory from those bone marrow cells to the in vivo cell types along the first principal component. But the trajectory that we're seeing for the pluripotent derived cells is coming from pluripotent stem cell derived endothelium, hemogenic endothelium, and the iPS cells themselves. So they're not patterning through the same developmental process as you would see in bone marrow. And we can further confirm that by projecting onto our atlas single cell data from a very recent study which looked at yolk sac derived, human yolk sac derived macrophage biology. And beautifully you can see in this yolk sac derived biology, very early stages, sitting with the erythroblasts of the blood atlas, and fetal microglia within our atlas sitting right on top of the later stage macrophages from that have been derived from yolk sac. But again, the trajectory is not explaining the behaviour of the pluripotent derived macrophages. Pluripotent derived macrophages are not mimicking fetal macrophage biology. They're something hybrid. And I'm not going to spend time in this talk explaining to you how they're different. We have got a paper on this on bioRxiv, and I'd be happy to answer questions about it in the panel. Just to summarise, I guess what I'm trying to say is that stem cell models of organogenesis are really important and very useful. And I think that the complexity of the organs that we can make in a dish were really beautifully exemplified by Barbara and Carl. But even very simple cellular models like the macrophage model, they are not perfect mimics of what's happening in the body. I also think that there is a really big gap in the single cell field around computational methods, and we need new methods to reveal genuine biological signal, particularly when the noise quotient in the data sets that we're working with are really big. And I would urge the Human Cell Atlas Asia group to really consider very seriously taking up the organoid models as an opportunity for discovery, capacity building and translation across HCA global populations. And I think this could address some really important equity issues that we're struggling with as a community now. Acknowledgements from me. I showed you the work of Nadia Rajab, very talented PhD student in my lab. The atlas was built by Paul Angel and Jani Choi here, and the single cell work was done in collaboration with Kim Ann Lecau and Yidi Den. Thanks very much.
Thanks, Christine. Some food for thought there. So let's invite all the panelists, which are the speakers, as well as welcoming Minoru Takasato from Riken in Kobe, Japan, who will join the panel. And Minoru is, do we have Minoru? Yes.
Yes, I'm here.
Yeah. Okay. Great. Whose work aims to create synthetic kidneys using organoid technology, which is really sort of the science fiction dream of this field, I suppose. So please go to slido.com, use the HCA dev code to submit questions and vote. We have some questions already. And while you're asking and voting, I would like to kick off with the first question that I'm going to direct to Minoru, which is, the question is, what are the challenges to scale organoid technologies into full solid organs, such as kidneys for transplantation? And I guess how far are we away from that becoming a reality?
Yeah, that's very important questions. In terms of the kidney organ, so I'm a researcher focusing on making a kidney organ from using the iPS cells or human ear cells. And I'm realizing the most difficulties for that question is the maturity and the complexity of the kidney tissues, how we can reproduce those things in the organoid as similar as the in vivo human kidney tissues. And I think that probably there is many answers for that question. But if I can pick up one very important factors for the maturating kidney organoid is the vascularization, I could say. So the vascularization is required for the not only the size of the organs to big, or the, but also the nutrient and oxygen supplies to mature the tissues in organoid, and which is we actually haven't had realized in kidney organoid in vitro.
Mm. So would you venture a guess? How far away are we from the first synthetic kidney transplant?
Yeah, so what we are now trying to is to vascularize the kidney organoid in vitro using like a microfluidic system. And this is the first step. And if we could succeed to do that, then we can make the organoid size to the bigger size. And I don't know how many years we need to complete that challenge, but I don't know, maybe 5 or 10 years.
Okay, sounds promising.
Yeah.
Carl, do you want to add to that?
I was laughing because 5 to 10 years is always the answer. So I think we're thinking about the same issues with our skin organoid model, and of course building vasculature into the organoids I think is going to be very important. With the skin system, we're kind of fortunate because we're thinking about ways that we can actually we think we can get the skin organoids to about mid-second trimester, like fetal skin, in vitro, and then by transplantation, we're seeing features of more adult-like skin after transplantation. So in the case of the skin tissue, and thinking about future translation, we might be able to maybe we don't need to fully mature the tissue in vitro. We can just get it to a certain point, and then after transplantation, it will continue to mature to an adult-like stage.
And I think what is extremely important for that then is to really have a quality control set in place that, for example, makes sure there are no remaining pluripotent cells in the tissues, or, I mean, because that could be detrimental if you transplant that, right? But I think in the case of skin, yeah, this is very promising actually. And if we, I mean, I think there are two different things. One is to be able to replace a full organ, which is a really big goal. And then you really need to grow the organ on a very big scale, and you need to prevent any necrotic core and all these kind of things. And then also you need really the right structure throughout the whole tissue. And the other thing is if you can replace parts of a tissue, especially if a tissue has kind of a repetitive structure, then this is much easier, of course, than if you really need the whole structure. I mean, for brain, it is to really replace big parts is yeah. I mean, that is much further away, I would say.
Yeah.
But to replace some motor neurons or something, that's a different thing.
Exactly, or like I was thinking of dopaminergic neurons. Specific populations, I think, are
yes, exactly.
much more tractable in the near term.
Yeah, I'm not sure.
Also I think.
Okay.
Also, their single cell analysis is, I think, has to be the standard way to assess how good the sample is before putting it into a human being.
Mm-hmm.
Yeah, I was going to say, I'm not sure I want to replace my whole brain. Maybe I should. But I know that there are ideas of using xenotransplantation as a step along the way. So you would transplant these, like Carl, you showed, you could grow human hair on nude mice. Would that be a way forward, or is there too many issues with having an animal host in between as a kind of a carrier for growing the organs?
Yeah, that's a potential way for it. I don't know. That might be problematic. You could think of for the skin tissue. I guess you could. The pig might be a good model to move into as kind of a carrier. That might be a hard sell for the patient population. To convince them to take the human graft off of a pig. But yeah, that's an interesting idea.
I guess there will be pig cells invading the tissue, and one has to somehow see how to deal with that.
Yeah, how would you do that?
If it's an immunocompetent animal, then immune cells for sure will invade the tissue.
Yeah.
How much kidney would you need to replace to get function back? Because presumably you're not looking at putting a whole kidney in.
So as the clinician said, we just need 10% of the whole kidney function to avoid the kidney dialysis. So we don't have to make the same size of the human kidney, but we just need to have 10%, only 10% size of the kidney for the patient. So I think it is good news for us, but still we have a very the challenging thing. We have to tackle like a maturation and a complexity. I think that's the same as the brain as well. Am I right, Barbara? I think maturity is very important, and I think skin needs some kind of UV light for the maturation. I just guessing, and the brain need to be, the learning for maturation as well. I'm not sure, but I guess.
Yes.
So another question from the audience, what are the disadvantages of organotypic cultures compared to organoid culture to faithfully recapitulate tissue? So the question is about comparing organotypic cultures on the one hand and organoid cultures, and what are the trade-offs there and advantages, disadvantages? Maybe this is for Barbara first.
Yeah, I mean, that's a good question. Of course, organotypic culture, I mean, you start at some time point where you get the tissue, and of course it limits you into what you can look at. And then one question is, how long can you keep that in culture actually? Do you have all the cues? Do you know what you need to give to the tissue in order to further mature it? So I think there's a limitation to how long keeping that in culture. During the time that you can keep it in culture, of course, you have a tissue that really has all the components that you need. But there is an advantage to growing all that in vitro, because you can maintain it longer, because from the beginning you provide it's kind of an iterative process to get the tissue better and better. But then once you are on that path, you know how to keep that in culture for a long time. So I mean, we can grow brain organoids for a year in culture. Of course, there might be a necrotic core that comes, but generally the tissue further develops. And if you get a organotypic culture from a fetal brain, I don't know how long you can maintain that.
Yeah, I think there is a discrepancy there, and maybe the organotypic cultures could learn something from the organoids in that sense. I think organotypic cultures were developed to the point where you can, at least in the brain, you can culture them for a few weeks. But it seems to me that if you can grow an organoid for a year, that should be possible to do better there with the organotypic culture. So maybe there is
well, actually, by doing both, you might actually learn what do you need in the media, what do you need to provide to primary cells such that they live longer. And that is also what, in principle, you need to give to the organoid in order to reach such a state. I mean, I think there's a great opportunity there. But yeah, and the other thing is, to go in and manipulate. Of course, you can throw viruses onto these the primary cells, but with iPS cells, you can really maintain them in culture, equip them with all kinds of different things, and then grow the tissue, which gives you a lot of freedom to look at mechanisms.
Yeah,
I agree with Barbara. In terms of kidney, we learn a lot from the organotypic culture of the mouse every kidney, and we apply that same method to the kidney organoid culture in vitro.
Yeah, I'll chime in and say I agree too. I think when referring back to our skin organoid model again, I think the skin has one of the richer histories of in vitro cultures. It's 40, 50 years ago, Howard Green's group first was able to grow or culture human keratinocytes in a dish. And it was really one of the first tissues that was widely used as an in vitro model. But it's been a consistent challenge to generate new appendages within these in vitro systems, and so now we're trying to use our success with the organoid model to go back and see if we can take primary cells and primary tissues and better culture them, using the culture medias that we've settled on.
Mm-hmm.
I have a related question for. I'm sorry. Go ahead, Christine.
I was just going to say it's probably obvious, but of course, as soon as you explant any tissue, it's no longer equivalent to what you had in the body. You have an element of de-differentiation and selection there as well. So there's always a compromise with these in vitro models. The point is that they're useful, and I think you choose the model that's going to get you furthest with the question you want to ask.
Mm. So a follow-up question then for you, Christine, from the audience is what are the computational challenges in comparing organoid with in vivo tissues? So that goes right to your question or your comment there. How do you distinguish biological differences that are true versus biological differences that are artifacts, and technical differences that are obviously artifacts?
I think the biggest challenge computationally is that we just lack good reference data sets to compare the organoid models with. So there are a lot of gaps in the reference developmental data sets that we have. And it is true that we can fill in quite a few of the time courses, developmental time courses, for example, by looking at what's happening in a dish over an equivalent time. but we don't have all of the structures and tissues and morphogenic gradients that are happening in that developmental tissue either. So they're not, it is a useful but imprecise map, I guess. I think the second big issue, of course, if you've got a reference that's missing a critical cell type, you aren't going to be able to identify it in your organoid either. And there's an assumption that the developmental maps that we have at the moment are capturing all of the tissues that are relevant, and we know there are selection biases in the way you disassociate them. And then the third challenge is that sometimes the cells that we make in a dish they don't have a developmental equivalent. They are a consequence of being an in vitro derived cell. And again, they tell us something really interesting about how genes and networks work, but they are hard to define in a strictly reference model.
Can I have something to say? Yeah.
Yeah, go ahead.
So yeah, I honestly, I try to I like the transplantation of the kidney organoid rather than the culture in vitro, because the transplantation makes the organoids much mature, and especially for the epithelial cells, after transplantation, the host mesenchymal cells greatly support the epithelial cells of the organoid to keep them healthy and more mature. So the comparison between the in vitro culture and in vivo culture is definitely very different, and I'm not sure how we can fix or normalize them when we do the single cell analysis. Maybe we have to do better to do the analysis after the transplantation of the organoid into the host. Something like that. I'm just thinking.
And if I may add something, so I think to really assess quantitatively how similar is an organoid cell to a primary cell, yeah, we have been trying to do that. We find it very difficult because, I mean, as Christine said, not all the reference has missing data sets. I mean, we miss time points, but it's difficult to assess which genes, for example, to include into such a comparison. And yeah, there are genes that are detected based on some technical artifacts, and then there are you can of course find marker genes in both the primary and the organoid data set, and then take the intersect. Or I mean, we experience that depending on which genes you include in this comparison, you really get different results. And at the end, it ends up being a relative comparison. So you compare your organoid cell to all the atlases that are there in principle, and you see what is it most similar to. And for that, it has been extremely helpful to really include a lot of different cell types, even cells from other tissues in this comparison to really say, okay, this is the most similar cell type. And I think there, once we have a really mostly complete primary atlas, I think this will be super helpful. Currently, we are still having kind of sparsely data sets here and there across development, and we can do the comparison. We are limited to that. But then once we find the most similar time point to really say, okay, it is 90% similar or whatever, this to me is very hard to do because of, it's unclear which genes to really include. In that case, it's helpful to have data, primary data from many individuals across many different, across many different methods, so that you know what is robustly detected, and the same you would need for organoid, and then really take these robust genes and do the comparison, I guess. I don't know. I'm not a computational person, so maybe someone computational has some idea how to do that really quantitatively.
I'm not a computational person, but I just wanted to say that I think that's a great point of including diversity of different tissues and different cells that have been analyzed using different platforms within your reference atlas. We're finding that in our comparisons to the reference data that we're wondering how well we can trust the harmonization of the reference in our organoid data sets, because it seems to be almost too good. And including
so it's designed to do.
Yeah, exactly. And then including additional references seems to be a good way to tease apart the real harmonization versus what's being just kind of an artifact of the computation.
And I guess it shouldn't, and I mean, then integrating data sets and having a 2D representation of primary and organoid together is very helpful and can visualize aspects, but then one shouldn't leave it with that, but really do DE within individual populations and then try to find out whether the differentially expressed genes give us any information on what is missing in the organoid, or is this all technically induced DE, or what? Yeah. So I think integration is great for showing it all at once, but we shouldn't end the comparison there.
So I agree with the idea of bringing lots of different cell types together and having them measured in many different ways, and that's exactly the motivation for the blood atlas that we brought together. I think the other point that lesson we can learn from other atlas projects is that in a single experiment, the observation you're making is anecdotal without a lot of experimental validation. If you're able to see it in additional data sets, it becomes a generalizable observation and you trust it more. If you've got enough scale, and in particular enough genetic diversity in the data you're looking at, not just replicates of the same cell lines again and again, then you can start to build rules of behavior. With the iPS-derived macrophages that we see, which are sitting quite a long way away from the in vivo macrophages, they're actually failing to switch off some of the mesenchymal developmental signals that they shouldn't have. So they're patterning all of the myeloid networks really beautifully, but they're just not silencing all of the mesenchyme networks they should. And that goes beyond whether or not they have seen a tissue context, but actually says something about how we're shortcutting. We haven't quite got the patterning right early in that developmental process.
Follow up there from the audience for you, Christine, or more specific question: could the lack of tissue environment around these cultures impact the resemblance of iPS-derived macrophages to the in vivo tissue macrophages? And if you could elaborate on that.
100%. Macrophages are great to study because they are completely responsive to their environment. But what I will say is that in the atlas, we did include macrophages which were iPS-derived and then had been put into humanized mice and taken from the lung, or had been co-cultured, for example, co-cultured with astrocytes or co-cultured with neurons for quite a long time, in some cases months, and that didn't make a difference. So it goes beyond the fact that they're not getting the right niche cues, and actually says that we are not deriving them in an identical way. And the network of genes that's pushing them out is a large set of extracellular matrix proteins which are not being silenced. So they're ectopically expressed in these cells.
So switching gears a bit, we have a question about the participation in the Organoid consortium. So how accessible is the activities and participation in the HCA Organoid consortium to people in groups based in Australia or Asia? And let's take that first part of the question first. I suppose that would be for Barbara.
Yeah, so I must say that this kind of organoid network is just starting. We like Grey Camp and my group, we have early on tried to push kind of that we should include organoids, and of course a lot of people are generating single cell data from organoids, and only more recently we thought one should kind of structure that and then also try to collect all the data and interact regularly. So there has not yet been any meeting of the organoid network, and it is well, then we thought, okay, who should kind of try to be coordinators? So then Christoph Bock and I came together, and we will now come up with a list of initial people and then initiate a meeting, and then we will go from there. And of course, people that want to be on this list, just let me know and write an email. And then these people will for sure know other people, and in this way the network will grow. So currently nothing has happened yet. So I think it should be as accessible to Asia, Australia as it is to US and Europe, because as you pointed out, Christine, there's tons of organoid work going on in Asia and Australia as well. So I think this is a great example, this Human Development Cell Atlas, where these meetings happen actually twice, such that it can reach all the people in the world. So we will need to do something like that, because people might not want to get up in the middle of the night. Yes.
And the second part of that question is whether you have created, or I guess will or plan to create a list of organoids being developed or worked on, or a registry of organoids. And specifically the question here is about pancreas, whether you know if anybody's working on pancreas organoids.
So actually, since the first of these seminars, the Europe US ones, people have contacted me and said, "Oh, we have some interesting data on, placenta development, on different aspects, and we are generating data." So I think we are currently gathering this information. I think there is work on pancreas organoids actually in Germany. There must be more throughout the world. So I think through that network, we will try to bring this together and then gather the data and make it available. Yes.
Mm-hmm. So, I know the many members of the RICAN in Japan. They are contributing to the HCA for the sequencing and single cell analysis, but not many for the organoid. And I'm actually not a part of the HCA as well. So, but I think I need to contribute to the HCA, and also I want to have information of the human cell atlas to make our organoid technology better.
I mean, we have been just submitting data to the DCP of the HCA, the data coordination platform. So I guess this is how you can contribute. And there's information about how to do that, and then the data will be publicly available. And then I think we should somehow gather the information, what kind of organs, what kind of time points, what protocols are available. Also, it is great to provide all the protocols that we are using on protocols.io, I think. To make also all that available.
Yeah, I was going to ask about that. Is that the best platform, the protocols.io? Is that?
I think that's what's proposed.
Okay.
By the HCA and the CCI. Sten, is that's right?
It's I think the most popular one right now.
Yes.
It is a commercial platform, but it's I think free for sort of public protocols, and then they charge you if you want to have personal protocols, or maybe they only charge companies. I don't know how it works really, but it's a very nice platform, and many people use it to share. There are channels, so there's an HCA channel with a lot of protocols for human cell atlas related activities, and many individual groups also have their own channels there. or rooms, or I forget what the name is now, but there are sections for different thematic sections, I think. And maybe we can then ask a related question about standardization, I guess. So if organoids were to be included in the HCA, how do you see it supporting multiple derivation techniques and protocols for the same cell types and tissues? Do you support, would you support all of them and just let users choose, or would you set some minimum standards, for example? That's the question.
I mean, currently the way it was, the metadata that you have to provide if you upload to the DCP is, well, that got somehow, when we uploaded organoid data, we thought what is actually useful to provide. And of course, in the case of organoids, there's a lot of additional things that you might, or other features that you might want to highlight to really provide all the information possible about which protocol you used and which line and which passage, even passage number of a line can make a difference. So I think, yeah, so this is also something that probably will be updated more and more the more people actually upload organoid data and think about a new metadata that might have to be provided.
Yeah, how do I know as a user of the data, if I go there to the portal and I find data for liver organoids, how do I know what that is? Can I trust that it's going to be liver? Is it going to be very similar to liver, just a little similar to liver? Like, what are the standards there? That seems to me a difficult question.
Yeah, we will need to think about that. I guess currently you can upload and say here, we have uploaded brain organoid data and then provided information on which line we used and which protocol we used and so on. But it's right, because sometimes even using the protocol, the organoid culture doesn't always perform well, and you get a lot of off targets. And then if you just say, well, I followed an organoid culture protocol, but there's actually a limited number of neurons in there, then it was maybe not a cerebral organoid that you really made. So we have to see whether we need to somehow do a quality control before including data.
Now, I've got a quick question about the DC.
I don't have enough information in this case. Yes.
Yeah, so I haven't done this yet. I haven't uploaded our data. Do I need to go upload specifically to DCP, or if we've uploaded to another platform like Gene Expression Omnibus, can they pull that information?
Yes, actually, okay. I think it can go either way. I think if you have already uploaded in the past, then it can be pulled, but you can also upload it there. And I don't know if you can then provide an accession number, or if then it can be in addition provided somewhere else. I don't have enough information about that. Stan, do you know whether uploading on the DCP and then providing some access to that is sufficient for example, publications?
Yeah, I don't know the specific directions that you can go with the data, but if you contact them, they are very happy to support you, and they will help you.
Actually, in the chat, I see that Mas said repository, for example, Geo, and then DCP for now. Okay.
Yeah. I know that they are working on connecting the European Genome Archive, which is where we, at least in Europe, submit controlled access human data, such that it can then flow from there to the DCP. And I think that's partly implemented, but there's maybe more work to be done there.
Can I put in another plug for DCP and just say, if you're a bioinformatician, the data wranglers there are super helpful as well. They really want people to use it. And so I would encourage everyone to contact them, because it's a really great resource.
Yes, absolutely. I agree. That's my experience as well. They're very helpful, and they will guide you.
Mm-hmm.
I have a question from the audience about ethics. where are we in terms of bioethics and public awareness of using human cell-derived organoids, specifically for cerebral organoids? And what can the Human Cell Atlas Organoids Network do to create standards and awareness? It sounds like a question for Barbara initially.
Well, I think, yeah, we can discuss that in the panel. I think, I mean, I think this is an active field. Actively, ethicists, regulatory bodies, you know, are discussing what is the ethics behind and what should be the regulations and the limitations of growing organoids. How far can they be matured? And of course, the ISSCR plays a big role there and has brought out, you know, has brought out frameworks for what one should consider and what one should follow also as a stem cell biologist. And I think in particular, of course, I mean, one type of organoid is, of course, the gastruloid, which really mimics early embryonic development and gastrulation. And there, I think the regulations are really that you can't mature that further than you could mature any primary kind of embryo culture. Which makes sense because at some point, of course, I mean, you are then dealing with a early human embryo. Even though it's in vitro, it actually, the better it grows, if it grows all the different germ layers, of course, it can give rise to some complex in vitro system that mimics an embryo. Yeah, so I think depending on which organoid you work with, this can be more or less problematic. With brain organoids, I guess the whole field tries to mature them further and get them grow more stereotypically. And currently, I would say there are, I don't really see big problems because they really mimic brain tissue very early on in development, and they are lacking a lot of factors such as vasculature, and of course it's not linked to any other organ system. But if you start linking things up and trying to mature further and further, of course you run into ethical problems.
Christine, Christine.
But I think currently the organoid does not think. I mean, it is not the neurons are not at all mature. They are very early developmental cell states. They start maybe to form you know, neuronal networks, and of course one wants to study neuronal networks, and people have shown that they can measure action potentials and can see that neurons are connected, that synapses form. You have inhibitory excitatory neurons, but currently this is nothing that really resembles a mature brain.
Yeah, I wanted to ask Christine if you put the ethics question in relation to your points about diversity and equity, and especially I'm thinking of the very different approaches that different cultures and countries have to these questions, where something might be perceived as very unethical in one place and almost imperative in another place. And if you would like to discuss that a bit.
Yeah, I mean, I think in terms of legalities and the regulatory aspect, you've got to work within a local community. I think whether an organoid has a moral standing is a question that is very personal. And that whereas, you know, are you dealing with this research under an ethical framework that's agreed by your discipline and by your government is a separate question. And then what are you allowed to do with that under community expectations, and the legal framework that's been evolved around that is really fraught, because often the community expects that we can do more than we can actually do in the lab. I think that really important point is that all of us as scientists have to be engaging with many conversations with the general public, with regulators, with stakeholders of our research. And although our scientific perspective is one valuable perspective, it is just one. And we really need to be extremely consultative about this if we want to move particularly things like the time for a gastroloid in culture, or the level of connectivity that you might have between sensory organs and a cortical organoid, then we can't do that in isolation. We have to come back to the community. That's painful because it's long and slow, but it's incredibly rewarding too. And I think we are often surprised by how willing the general community are to support what we're doing, particularly in the stem cell field as opposed to other technologies.
I think that's a really great point, Christine. Yeah, it's a conversation, and I think it's a conversation that clearly people are having as we develop these more and more complex systems, and we just need to keep the communication lines open, and keep having these conversations to determine where the lines should be drawn as we move forward. Mm-hmm. Yeah, it's certainly something we're thinking about in our work with these sensory systems now, as we move to trying to connect them to their central targets. We see pain sensory neurons, touch sensory neurons within our organoids. Where should we be drawing the line as we build these systems and make them more complex?
Yeah.
And I think, yeah, you go.
Sorry. So in terms of ethics, I think the conclusion from each community could be different. Of course, it depends on the religions as well. So I'm just wondering which conclusion we need to follow. Maybe I just follow the conclusion in Japan, but the science is the global, not the local. So we're just wondering which conclusion we should follow. Maybe Christine, you have some idea.
We as a discipline have to have a position on this, and I think that's why the role of ISSCR is so important here, because ISSCR actually brings some very, very multidisciplinary perspective to this problem. But that's only one part of the equation. I think with you need to work within your institutional ethics framework. You need to work within your own moral compass. You would never force someone to do something that they felt uncomfortable with because it's outside of their cultural or religious boundaries. So there are many, it's a very, very layered and nuanced answer, Minoru.
All right, everybody. We are going to have to wrap up. This was a great discussion and a great session overall. I want to thank all of you for your contributions. We have a few pieces of information that we want to share before we end. First of all, thanks to the program committee that puts together this seminar series. Bruce, Gary, Alain, Dana, Moose, Aviv, Diane, Sarah, and of course the fabulous HCA Executive Office. We want to thank the panel, Christine, Minoru, Barbara, Carl, for the whole discussion and the talks. And we are happy to announce that the next Human Cell Atlas Developmental Seminar will be on neurons, my favorite cells. And I will be speaking along with Frida Miller. And the date, put it in your calendars, is October 27. And there will be a follow-up as usual to be determined for the Asia and Australia. You can go to the humancellatlas.org webpage to find more information on this seminar series. And you can also find information there about the biological networks and the project registry and news items and so on. And you can also follow us on Twitter for continuous updates. Today's recording will be posted on YouTube. You will have to quickly memorize this very long URL. I'm not sure that's going to work, but or you can search for, just go to the Human Cell Atlas channel, I suppose. And then, as always, we want your feedback. So please take the feedback survey, which is in Slido. And you can either go to that address there, or you can take a picture of the QR code to get there. We have a number of additional events coming up. The Human Cell Atlas Latin America workshop on September 24th and 25th. A very timely COVID-19 symposium, don't miss it, October 1 and October 9. And there is an HCA Asia virtual meeting as well on October 22-23. And I think that is all for today. So thank you, everybody. Bye-bye.
Thanks.